In this lecture, or this PowerPoint, we're going to start with going through what are some basics of microbial metabolism. Just getting through the basics in this introduction. And so the overview of the entire PowerPoint includes what are some basics of metabolism, including what is metabolism, what are some chemical reactions that are happening, how do enzymes play a role in metabolism, and then we will finish out in lecture how we actually break down carbohydrates, what's happening in anabolism, as well as how we can regulate all of these different types of chemical reactions. So just in this little introductory video, audio, we're going to go through kind of the first three bullets here. Metabolism basics, chemical reactions, and enzymes. And so since we are going to be talking about metabolism, and you may have heard the word metabolism used outside science classes when people talk about, I have a fast metabolism or a slow metabolism. Well, the actual definition of a metabolism is a collection of controlled biochemical reactions that take place within a microbe, which is a mouthful all in itself. But really what it's talking about is all the chemical reactions that are happening. Anytime anything is getting broken down, anytime anything is getting built up. And so when people talk about having a fast metabolism or a slow metabolism, really what they're talking about, whether they know it or not, is all those chemical reactions that are happening, are they happening very quickly and efficiently, or are they happening a little slower and not as efficiently? Now, the ultimate function of metabolism, by breaking everything down and building things up, ultimate function is reproduction. All of these microbes that we're going to be talking about, yes, they want to break things down, they want to build things up, but their ultimate goal is so that one organism can become two, and two become four, and so on. Now, as we start talking about all of these different types of chemical reactions, there really are two types of chemical reactions. We call them in classes. They're kind of two basic ideas of what type of chemical reactions are happening. The first type of chemical reaction or class of chemical reactions are called catabolic reactions or catabolic pathways. What's happening in catabolic pathways, or sometimes you just hear catabolism, is that it's taking large molecules and breaking them down into smaller molecules. So it's just breaking things down. Now, when we break things down, that releases energy. It's an exergonic reaction. Exo, or X, means release. And so when we break things down, we release energy. And yes, for many organisms, including ourselves, we can use that and make ATP. The opposite then of a catabolic reaction, instead of breaking things down, is we can make or synthesize large molecules from smaller molecules. Now, a lot of times, the smaller molecules came from catabolism. That as something got broken down, it made these little smaller molecules. I know the size doesn't look different than this over here, but they're smaller. They make these smaller molecules, and we can take these smaller molecules and we can make larger molecules through an anabolism or an anabolic reaction. Now, when we build something, meaning we're making it larger, that requires, it's supposed to say require. That requires energy. Change that to requires. I'll have to fix that. Which makes sense. If you are building something, that requires energy. If you're going to build a building, or do any type of building of whatever, that requires energy on your part to build it. For something to get broken down, it doesn't take much energy and things can fall apart. I'm like, my kind of tip for remembering which reaction is building, which one is breaking. If anyone has ever owned a cat or knows someone that owns a cat, cats like to break things all the time. They're knocking things over, they are breaking things. So I always like to think cats like to break things. Catabolic reactions are breaking molecules down, which, yes, does require or release energy. Now, this big, huge diagram you're going to see again, because when we're talking about metabolism and all of these chemical reactions, breaking things down, building things up, all of these other little steps, and I'm like, this is all metabolism. It's all of these different types of chemical reactions. And there are 8 basic statements that happen in metabolism. Now, the first one, and I wrote them down on my next slide too, and I'm like, so I'll kind of go back and forth, is that my first little statement here is that yes, every cell, and some organisms are only one cell, every cell has to acquire nutrients. And I'm like, for us to break anything down, stuff has to first go in. And I'm like, so every cell requires nutrients. And I'll put these up in a second on my next slide so you can write them all down and you can pause me. And I'm like, the second step is that metabolism, so all these chemical reactions, requires energy from either light or the catabolism or breakdown of nutrients. And so for anything else to happen over here, we need energy. And for us, we're not photosynthetic, so we don't get energy from light. We get all our energy by breaking down these nutrients. Now, this is the same thing on a bacterial scale, same thing on any small cellular scale. This happens in us too. For us to do anything, we need energy from nutrients and it has to get broken down. The third statement is that most of this energy that gets released from breaking down these nutrients is stored in ATP. The next step, or the next statement of metabolism, is that cells will catabolize, break down, nutrients to form these precursor molecules. So yeah, we release energy, but also by breaking things down, we also make these smaller molecules, and we call them precursor because it's these smaller molecules that can be used shortly in building. And so that gets into our anabolism. We can take these precursor molecules, we can take the energy that was released from breaking things down, we can put it together. We have molecules, we have energy, we can build things. And we can create, or the organism can create whatever it needs. The sixth statement is that enzymes, and we'll talk more about enzymes, plus energy will start to take larger and larger molecules and make them bigger and bigger. So it will take larger building blocks and make them even bigger, and we call them macromolecules. And I'm like, it's just assembling everything together. The seventh statement is that yes, cells are going to start to grow by assembling all of these macromolecules. Now, and I'm like, we can take some of these macromolecules and we're going to actually see them as cellular structures. They get large enough. And we can take some of these cellular structures and we can actually start to see our cell grow. And I'm like, we have more organelles, we have more substrate to the cell. And then eventually, as this little arrow is leading to, as soon as the organism has doubled in size, and I'm like, it will reproduce. And I'm like, that's its main goal. It wants to become bigger and bigger and bigger, make all the cellular structures it possibly needs, and then it divides. The ultimate goal of metabolism is to reproduce. Now, yes, these organisms reproduce differently than us. There's no mitosis happening and there's no meiosis happening. They go through what's known as binary fission. Bi means two, fission means split. They split into two, and now one became two. Now, here's all of my 8 statements of metabolism. If you want to write them down, you can pause me right now. Otherwise, I'm going to continue on into a couple other different types of reactions. Other than just building and breaking to make large molecules and to break large molecules down into small ones, when that happens, when we are breaking things down and building things up, energy is not always just transferred around and stored as ATP. Unfortunately, not unfortunately, and I'm like, one of the things that we can move energy around is the movement of electrons. And so when things are getting broken down and when things are getting built, and I'm like, we're also looking to see is where are the electrons? Because electrons carry energy. And so the basics of these reaction is that, yes, electrons and their energy will come from some type of electron donor, and it will eventually go to some type of electron acceptor. And so energy is transferring from a donor to an acceptor. Now, reactions always occur simultaneously. If something loses an electron, something will gain an electron. Now, and I'm like, my little reaction here. So we are making A and B, we are mixing them together, and they will become my products of A and B. Yes, they're the same name. However, what happened is that one of these molecules, A, will lose an electron. And I'm like, so during this reaction, A loses an electron. Now, that electron has energy, and that electron, so A becomes this, will actually transfer over to the B. So B gains the electron. So A lost the electron, it was the donor. B gained the electron, it was the acceptor. And so my little reminder, my little review, if you remember, what charge does an electron have? Well, electrons have a negative charge. And so when A lost an electron, it lost that negativity and it became positive. And B, it gained an electron, and so it is now more negative. It gained something that was negative. I like to kind of think this if we refer to, what kind of friends do you hang out with? If you start hanging out with lots and lots of negative individuals, so if you're gaining negative friends, you start to become negative. Now, if you lose some negative friends, you just told them, I don't want to hang out with you anymore, you become more positive because you lost those negative friends. Now, and I'm like, the reaction when we're talking about losing an electron and which of the reactions or which of the substrates or reactants I'm like is gaining the electron, we call these oxidation reactions and reduction reactions. In the case of A lost an electron. That's called oxidation. That it lost an electron somewhere and gave it to something else. That's oxidation. Reduction is when some type of molecule gains an electron. Now, it's called reduction, and that confuses a lot of people. You're like, but you gained something, but it's called reduction. And people think it's an oxymoron. It's not, actually. Because when you think about what you're gaining, you are gaining something, but you're gaining something that's a negative charge. And so you actually have a reduced charge. And I'm like, you are more negative. You've reduced your charge. And that's where they get the reduction from. And I'm like, oxidation is you're losing an electron, you become positive. Now, my kind of little way to remember the difference, and I'm like, I didn't come up with it, but if you can remember the acronym Oilrig, like where they go drill for oil. Oil can stand for oxidation is loss of an electron. Reduction is gaining an electron. Now, electrons generally just don't float around all that often, and like, again, usually as one electron has lost, something else is picking it up. Now, there are some molecules that pick up electrons very quickly, very easily, and can transport them. Now, as you transport an electron, you are transporting energy. Now, like we call these electron carriers. That's their job. One of the most common electron carriers, and I'm like, is usually anything that has a hydrogen atom in it. And the three most important electron carriers in most cells are NAD, NADP, and FAD. When it picks up an electron, it usually picks it up with a hydrogen, and so NAD becomes NADH. That H is showing that it has picked up a hydrogen as well as an additional electron. That additional electron made it lose that positive charge. NADP, when it picks up an electron, usually tagged on to a hydrogen, and so you've got a hydrogen plus the extra electron lost its negative charge. Or FAD, and I might can become FADH2. It picks up two hydrogen, two electrons, and I'm like, and the charges will equal out. Now, I don't care what the NADs and NADHs stand for. I just want you to be familiar with NADHs, NADPH, and FADH2, as these are electron carriers, and as they carry electrons, they are carrying energy, which is going to be used later. Now, because not all energy is stored or carried around as electron carriers, and I'm like, some of this energy has to get put somewhere else. Now, catabolic pathways, again, anything cats break things, and I'm like, as molecules are getting broken down, as this energy is getting released, it has to get put somewhere. And so a lot of times the energy that's released by breaking things down is concentrated and stored in high energy bonds. Now, one of the most common places that we can store energy in high energy bonds is in the molecule called ATP. Now, if you remember what ATP stands for, adenosine triphosphates, this is your ATP. And like there's your the adenine, which makes up the adenosine. These are your 3 phosphate groups. It is these molecules, and I'm like, or these bonds right in between each of the phosphates. This is your high energy bonds. Now, and I'm like, as we break molecules down, that energy that is released gets used and through a process called phosphorylation. It just means we are adding phosphate onto a substrate. These 3 phosphates were not always there. Most likely there was only 2 of them, and sometimes only 1. I'm like, however, if we have extra energy, we can use that extra energy and shove a phosphate group on here, making this high energy bond. It's called phosphorylation. We're just adding a phosphate group on. I'm like, now, and I'm like, it's those high energy bonds that, yes, later we can break them down and use it anywhere else. And so, such as our anabolic reactions. Anytime we want to build anything, we can use the energy of ATP by breaking these high energy bonds, releasing the energy, including the electrons, and using it to make whatever it is we need to make. So it's this constant getting energy from catabolism, making ATP. And then anytime we need to build anything in anabolism or in anabolic pathways, we break down the ATP. We usually remove 1 phosphate group, and ATP becomes ADP, the adenosine diphosphate. Yes, on occasion we can remove 2 phosphate groups, and it becomes AMP for adenosine monophosphate. But it's this constant breaking things down, releasing energy. Yes, we're going to make ATP or break ATP down and use that energy. Now, enough away from energy. We're going to get back to energy later. But, and I'm like, with all of these different types of breaking things down and building things up, proteins come into play. So, my little review of proteins, some basics. I put a lot of different pictures up here to kind of jog your memory of proteins from A&P classes. But some basics. One, and I'm like, proteins have several different functions. Some functions of proteins, one, we can find them as structures. I mean, a lot of your muscle is made up of protein. We can find enzymes there, or we can use proteins as transportation. On the cell membrane, we use protein carriers all the time. They can be used in defense. They can be used in regulation, moving things in and out of wherever it needs to go. And they can be used as enzymes. Now, it's that enzyme function that we're going to talk about, because enzymes are proteins. But yes, they're a function. Now, proteins are made up of amino acids. Now, my little review of the structure of amino acid, basic structure of an amino acid is right here. It has a carbon, and then that carbon can attach to 4 things. One of the things is an amino group. One of the things is a carboxyl group. One of the things is a single hydrogen. And this is standard. Amino group, carboxyl group, hydrogen. Amino group, carboxyl group, hydrogen. That's standard for every amino acid. It's just this R group that can change. There are 20 amino acids, and they all change based on that R group. Glycine's the simplest. It just has a single hydrogen. Other amino acids, other of the 20 amino acids, have a lot larger structures for that different R group. And I'm like, but altogether, 20 different amino acids, very similar structures. Now, again, the main reason why I'm bringing up proteins is because of the enzyme function. Now, enzymes are proteins. So what are enzymes? Enzymes are proteins. Yay, that's what they are. However, we care really about what enzymes can do for us. Now, enzymes are organic catalysts. Organic because they're carbon-based. And what a catalyst means, a catalyst is something that increases the likelihood of a reaction. Means reactions can happen easier, which means they're more likely to happen. Now, how the catalyst works, how the catalyst increases the likelihood of a reaction happening, is it lowers the amount of activation energy, it's a term, needed to start a reaction. Now, I've got a couple different pictures here. This first one is trying to show you I have reactants and I have products. And so my reactants will eventually become products. And even though as I break a molecule down, my molecule to begin with is called AB, it's smooshed together, I'm going to break it down into its individual components, A and B. I'm breaking things down, I'm going to release energy. However, to go from these reactants to these products, even though I'm going to release energy, to even start the reaction, it requires energy to start it. That energy that's required to start any type of chemical reaction is called activation energy. You have to put some energy in to get something out. Now, and I'm like, activation energy without an enzyme. That means for these reactants to become these products, I have to put this much energy in. That's a lot of energy, which means if there's not that much energy around, this reaction might not happen. However, if you have an enzyme that lowers the amount of activation energy needed, now we only need this much energy versus this much energy. If you don't need as much energy to start a reaction, it's more likely to happen. You probably have this little bit of energy around, whereas you might not have had this much energy around. And so catalysts increase the chance of reactions happening because they reduce that amount of activation energy needed. My other diagram that I like has to do with frogs. And so it looks nothing scientific really whatsoever. You have reactant frogs and you have product frog. And for the reactants to become products, they have to get over this activation energy barrier, which means they have to jump really high. And some frogs might not be able to jump that high. They don't have that much energy, which means they will not become products. However, what a catalyst does is it lowers that barrier, which means it is more likely that those frogs are going to be able to jump over and the reactants will become products. Now, my other little notes on enzymes. One, yes, they are proteins, and all enzymes have an active site. So they are a molecule. Most proteins are shown as like usually big purple or blue blobs. So here's your protein. It's a big blue blob. It's an enzyme because enzymes are proteins. And they do have an active site. In this case, it has almost like this two little section active site, but it's this active site that will bind to a substrate or some type of reactant. Now, it's very specific. This enzyme is only going to fit this substrate. Nothing else is going to fit. This is the only substrate that can fit in this active site. So enzymes have specificity. And a lot of times the enzymes are named for what substrate can fit. Now, once a substrate binds to the enzyme, it usually starts to form a more perfect fit. So it fits, but once it binds, it forms a more perfect fit. If you notice, this is kind of flat on the bottom, this is kind of round. Yes, this will fit in there. Not perfect, but as soon as it does fit in there, if you notice that, yes, that protein will change and it forms a more perfect fit. And so it's not going to be able to release itself. Now, and I'm like, this is just showing what enzymes can do. Now, my little notes on enzymes. Yes, enzymes are not used up. They are not changed. They are only recycled. They're specific for only one substrate. That's that specificity. And they will drive a reaction one way, which means they're either going to do some type of catabolism or anabolism, but then another enzyme would be needed to do the reverse. Now, an example, and I'm like, is just showing, here's your big this bluish blob. This enzyme's name is fructose 1,6-biphosphate aldolase. You do not need to know that name. However, my little note with enzymes, about 99% of all enzymes end in ase. So if you see something that ends in ase, that should be a big clue. That's an enzyme. Now, yes, enzymes are normally named for the substrate they work with. So this enzyme, fructose 1,6-biphosphate aldolase, is because the substrate is called fructose 1,6-biphosphate. Now, as this substrate fits into the active site, and there's starting to fit in, yes, it will form a more perfect shape. It's kind of rounded, now it's a little more specific for its shape. And what this particular enzyme is doing to fructose, and I'm like, is splitting it up. And so it takes this big huge molecule and splits it up into these two different molecules. I don't care that you know the names. But if you notice, I'm like, its job, take this molecule, break it down, releases those two products, and then it goes right back to normal again. Because it's not used up, it's not changed, it just keeps getting recycled. And it's ready right there again for another substrate, another fructose that it will break down into these two products. Now, because this enzyme is breaking molecules down, this is considered a catabolic reaction. It's breaking things down. Now, there we go. And I'm like, there are some things that can influence how well an enzyme works. And I'm like, temperature, pH, the enzyme substrate concentration, so how much substrate is there, and if there's an inhibitor or not, which means it's going to either block an enzyme active site or it's going to somehow change the enzyme so it can't break its substrate down. Now, this is just showing some of these factors that affect enzyme activity. An example, and I'm like, all enzymes seem to have, a very specific temperature or pH that they work best. An example, I'm like, this particular enzyme works best at around 37 degrees Celsius, give or take. That's body temperature. And most enzymes that are in our bodies, yes, work best at body temperature. A lot of microbes, including bacteria, their enzymes also work at best at body temperature because they would like to live in us. This is showing that, yes, this particular enzyme's optimal pH, and I'm like, is right around neutral, right around 7. And I'm like, it works best at that. This particular one is showing as that you add more substrate, fructose in that one example. If you add more substrate, the enzyme activity will go up. However, there is a saturation point where you can't add any more substrate because there isn't any more enzyme. An enzyme can only break down or build as much as it can. And I'm like, that the more you add to it, it's busy, it's doing something, it can't do anything else. And this is showing just, based on temperature and a couple different enzymes that, yes, although this is the optimal temperature for this particular enzyme in a human, right around 40 degrees Celsius. And I'm like, there are other organisms that live in hot springs that their optimal temperature is 70 degrees Celsius. That's extremely hot for us. And I'm like, but if that's where you live, is in a hot environment, your enzymes are going to work best at that particular temperature. So it can vary. Most of the organisms we deal with, and I'm like, optimal factors that affect enzymes are normally body, whatever our body is, body temperature, body pH. Now, when I talked about one of the things that can affect enzymes was the presence of an inhibitor or not. I'm like, here's the big blue blob, here's your enzyme. And I'm like, this particular enzyme, its active site is to fit this substrate. And I'm like, this substrate should be fitting right in here. However, you may have an inhibitor around. It's called a competitive inhibitor. That this inhibitor can fit in there too. It doesn't look exactly like the substrate, but it can still fit in the active site. Now, because it's occupying that active site, this substrate can't fit there, which means the enzyme can't do whatever it's supposed to do with that substrate. So it's called competitive inhibition. And I'm like, there's just something else that's competing for that active site space. And yes, no product is going to be formed because the substrate can't get broken down or built up, whatever it's supposed to happen. There's another type of inhibitor called non-competitive inhibition. It's called non-competitive, it's still an inhibitor, but it's not competing for the active site. Instead, you can have an inhibitor that binds somewhere else on the protein. So it's not up in the active site. Instead, it can bind at a site called an allosteric site. It is usually the opposite side of the active site. And as soon as that inhibitor binds there, I'm like, the protein or enzyme changes shape. And if you notice, I'm like, on this particular one, this little top part folded over on either side. And so by having this inhibitor here, and I'm like, the enzyme changes shape and the substrate can't fit there anymore. So it's called non-competitive inhibition. It's not competing for the active site. However, the end result is the same. Substrate can't fit, no product is made. And so 2 types of inhibitor. Competitive, it's competing for active site. Non-competitive, and I'm like, because it doesn't compete for the active site, but it still will stop substrates from binding there. Now, what a lot of times happens in microbes and in humans, and I'm like, is an end product, so whatever the ultimate goal is, whatever we're trying to make, actually can become one of these non-competitive inhibitors. My little example, the initial substrate is an orange circle. And this enzyme will take little orange circles and they will spit out little yellow triangles. That's the job of enzyme 1. Well, enzyme 2 is like, great, I love little yellow triangles. Let me take it and I will spit out little red squares, whatever it is. And then the next enzyme's like, awesome, I love little red squares. Let me take it and then spit out these little green ovals. And whatever the microbes, the microbe, whatever it wants to make, and I'm like, its end goal was, I need to make more of these. I need to make more of these little green ovals. Now, what can happen though is at some point, we only need to make, or microbes only need to make so many of these little green ovals, whatever they are. And I'm like, we made the green oval, let's stop making the green oval. Let's stop making more green ovals. And so an end product can actually go back into this first enzyme, fit in, and change the shape. It's a non-competitive inhibition. It's not competing for the active site, but it changes the shape of enzyme. And now enzyme 1 can no longer take orange circles and spit out little yellow triangles. And so we stop making end product. This happens in the human body quite often when making various molecules. It's like, all right, well, let's make, sometimes there's just various types of hormones. Let's make this type of hormone and release it. Well, at some point, we only want to make so much of that hormone. And so there's a feedback inhibition. That hormone will actually go back and, stop the whole process so that you only make as much as you need at a time. Now, and as if you're looking ahead, the next slide starts to talk about carbohydrate catabolism. This is we're going to get into in lecture. It really is breaking down carbohydrates. That's what carbohydrate catabolism is. And I'm like, it all goes together, so we're going to end right here and do the rest in lecture.